All right, Unit 2, Cell Structure and Function, and this is video number 4, Tonicity and Osmoregulation, which covers Unit 2.8 of AP Biology. So at the heart of this concept is understanding why water flows. So the process of water diffusing is called osmosis. So let's take a look at this diagram. What is the net direction of water flow in this diagram? And why are there black arrows shown pointing in the opposite directions? So we've got a big pink arrow and then a bunch of little black arrows. What do all of those represent? So that big pink arrow is showing the direction of net water flow. So overall, more water is flowing to the right than it is to the left. But that doesn't mean there's no movement to the left at all. It just means that there is less movement to the left than there is to the right. And if you count up all of those arrows, you'll notice that in this diagram, they're showing 1, 2, 3, 4 moving to the right and 1, 2, 3 to the left, so that overall there's a net movement to the right. In determining where water flows, there are two major contributing factors. There's solute and there's pressure. So we're going to talk about solute first. So what we have in this diagram is showing low solute concentration on the left and high solute concentration on the right. They're the same number of molecules on each side. So why is it that water goes towards the area with the high solute concentration? And the reason water goes towards that area with the high solute concentration is you can think of it as those solute molecules are kind of occupying some of the water molecules. So those solute molecules may be sodium ions or chloride ions are attracting some of those really polar water molecules. And so those polar water molecules are kind of occupied, they're kind of busy. And so we have fewer free water molecules left. So then if you look overall, we have more free water molecules on the left side than we do on the right side. And so then we have a net movement of water molecules from an area of high water concentration to low water concentration. So just like anything else, water moves from where there's a lot of water to where there's less water. But in this case, we really have to think of it in terms of free water molecules, not just the total number of water molecules. All right, so let's make some predictions. And to make these predictions, incorporate the terms isotonic, hypotonic, hypertonic, and see which way do you think the water is going to move in each of these conditions. So on the left, we have a sort of a medium amount of solute. In the middle, we have a low amount of solute, and on the right, we have a high amount of solute. Now, because I told you to incorporate the terms isotonic, hypotonic, and hypertonic, with this diagram, we can assume that those terms each apply to one of these solutions. So the middle amount of solute we can assume is the isotonic. That means it's going to have the same solute concentration inside the cell and outside the cell. In the middle picture, we only have a couple of solute molecules drawn. So that is hypotonic. So that's a solution that has a lower molarity. And then on the right, we have hypertonic. So that's a solution that has a lot of solute dissolved in it. So hyper means above, hypo means below, and iso means the same. So which way in each of these cases is the water going to flow? Well, if we have an isotonic solution, the water is, there's going to be no net water movement because the same amount of water is going to come into the cell as leave the cell. If we have a hypotonic solution, that's very dilute, a low concentration, there are going to be more free water molecules on the outside of the cell, which means the water is going to flow in and the cell will swell. Oppositely, if we have a hypertonic solution with a whole lot of solute on the outside of the cell, there are really very few free water molecules. And so what's going to happen then is the water is going to flow from the cell into the solution, which will shrink the cell. So let's make some predictions about Elodia. Elodia is a water plant or an aquatic plant. So let's try to figure out what might happen if you put Elodia into the following solutions. What if we put it into distilled water? And what if we put it into a 10% sodium chloride or table salt solution? And 10% sodium chloride by mass is a fairly high solute concentration for reference. So in the distilled water, which is a hypotonic solution, the water is going to flow into the cells. So here we're looking at a picture of the cells of the Elodia under a microscope, and you can see that they're really swollen, right? It's because the water flowed in. Underneath that, in the lower diagram, this is when the Elodia was soaked in a 10% NaCl solution. And so in this case, the water flowed out. So we still have the cell walls, so there's still that structure on the rigid structure of the cell walls, but inside of that you can see that the membranes have kind of shrunk down. It also looks like a darker color because those chloroplasts, which are green, are now in a sort of tighter space inside of each cell, so it looks like a darker overall color. All right, let's get another example of how water moves when we have solute. So let's imagine this is called a Utube, and so we have a semi-permeable membrane connecting two sides of this tube. Let's imagine that this semi-permeable membrane is permeable to water, but is not permeable to glucose. So we have C6H12O6, that's representing sugar. So we put some sugar in the left side. What do you predict is going to happen? So what's going to happen is because we have a high solute concentration on the left, that means water is going to flow into that area. So our net water movement will be from the right side to the left side. And remember that semi-permeable membrane allows water to pass but doesn't allow sugar to pass. So the water level is going to rise. So we know the water is going to move from the right to the left and the water level will rise on the left, but knowing exactly how much movement there is going to be important in this unit. So we're going to quantify that, which means instead of just figuring out which direction it's going to go, we're going to numerically quantify sort of the force at which it moves to the left. And to do that, we need to calculate solute potential. All right, so there's a lot of formulas stuff going on this page, but realize this is all on your AP Bio formula sheet, so you don't need to memorize this formula. You do need to understand what it means, though. So first of all, let's look at that symbol. There's that symbol for potential, and then it's sub S. So this is showing the symbol for solute potential. Like I said earlier, there are two big factors determining where water is going to flow. There's solutes and pressure. We'll get to that in a little bit, but let's concentrate on solute to begin with. So the formula for solute potential is negative I CRT. Now let's take a look at each one of the terms of that equation. I stands for the ionization constant. This is how many molecules does it break up into when one molecule of solute enters water. So for glucose, if you put one molecule of glucose in water, it will stay as one molecule of glucose because all of the bonds connecting the atoms of glucose are covalent bonds. And so it's not going to break apart into multiple pieces. It's just going to stay as one. So in that case, our ionization constant would be 1. If we have sodium chloride, NaCl, that's ionically connected. And so in that case, it's going to break up into sodium ion and chloride ion. So in that case, our ionization constant would be 2. All right, let's go on to that next one. C stands for molar concentration. So that's molarity. You know all about that. That measures how many molecules of the solute we have per liter. Okay, so next up we have pressure constant. You don't need to memorize this. This is listed on your formula sheet. And this is just kind of a way numerically to convert all of these other factors from measures of concentration into a measure of pressure, essentially. Okay, temperature is also going to play a role in this, so we need to know the temperature of our solution. The temperature of our solution in this equation is measured in Kelvin. Kelvin is very similar to Celsius. It just has a different, each degree is the same amount, it just has a different set point, which means that absolute 0 on a scale in Kelvin is where there's absolutely no atomic movement. So to figure out a Kelvin degree, you just take the degrees Celsius of the solution and add 273. And once again, this is on your formula sheet. All right, so let's do a sample calculation. What would be the solute potential of a 3 molar solution of glucose at 22 degrees Celsius under standard atmospheric conditions? And standard atmospheric conditions just means that we're allowed to use that pressure constant that's provided, which will always be true in this class. All right, so give this a try. Okay, so what we do is we plug in all these numbers. Remember, the formula for solute potential is negative I CRT. I, in this case, because it's glucose, is 1 because it doesn't ionize. Glucose doesn't break up into ions. The concentration is 3 molar. That was provided. The pressure constant is provided. That's the 0.0831. And then to find our degrees Kelvin, we take 22 plus 273 to find the temperature. And if you multiply these all together, a bunch of these units canceled out. We're left with bar, which is a measure of pressure, essentially. And so we have a calculation of negative 73.54 bar. Let's take a look and figure out what in the world that number might mean. What is that useful for? So to figure out how we can understand that number that we came up with on the last page, let's go back to this sort of Utube example. And that's U, the letter, not Y-O-U. All right, so what is the solute potential of the sugar? Well, if it's a 3 molar concentration of sugar, which is really pretty high, under the conditions that we listed on that previous slide, we end up with a solute potential of negative 73.54 bar. All right, so that's telling us something about that water. And let's look and compare that to distilled water, just plain water. So on the right side, we have plain water. So what's the solute potential of pure water? Well, we can figure this out from the negative I CRT equation. And it actually isn't a whole lot of math, it's just some thinking. So the solute concentration of pure water is 0, which means that C value in that equation is going to be 0. And if we multiply anything by 0, we get 0, which means that the solute potential of pure water is 0. Okay, so now we can look at this and say the concentration of pure water is 0, and therefore the solute potential is 0 bar. Let's just look at this and kind of take note. So if we have 0 bar and negative 73.5 bar, the water is flowing towards that negative bar, that negative solute potential. And this is important to keep in mind, is that water flows from high to low. So water flows from a high water potential to a low water potential. But pure water is 0. So what you really have going is water is flowing from a 0 potential to a negative. So it's going from higher to lower, but it's all relative. All right, so now let's take a look at this next question. Without adding any more solute, what could we do to make the water levels even again? So one thing we could do to make the water levels even again is push it back the other way. So we could apply some pressure. Well, now how much pressure would we need to add? We would need to apply enough pressure to offset the solute potential. So if you push with a force of 73.54 bar, which is the sort of opposite equivalent of that solute potential, your water levels will become equal again. Remember how I said that there are 2 main things that affect where water flows, solute and pressure. So here you can see both factors at play. Is the solute is trying to push the water to the left, but the plunger is pushing the water to the right. And if those forces are exactly equal, we'll end up with those water lines being exactly equal. So water potential is going to tell you which way water moves. If we have a membrane and 2 liquids on opposite sides of that membrane, we can figure out the water potential of both sides and then figure out which way the water is going to move. Reminder that the water is always moving from the higher water potential to a lower water potential. To figure out water potential, we need to know solute and we need to know pressure. So let's take a look one more time at this example, where on the left we have a solute potential of negative 73.54 bar, and we have a pressure potential of positive 73.54. So those cancel each other out to make an overall water potential of 0. That balances the potential we see on the right side. On the right side, there's nothing pushing it down, so the pressure potential is 0. This is always true of an open container. If it's an open container, the pressure potential is 0. So assume if it's in a beaker or just a jar or something, it has a pressure potential of 0. The solute potential on the right side is also 0, and that's because it's plain water. If there's no solute, there's 0 concentration, which means it's a 0 solute potential. So in this case, the water potentials are equal, and so there's no net flow. Water is going to go in both directions in the same amount, so there's no overall net flow. Because water potential problems can be kind of tricky, I'm going to give you a practice problem to think about here. So let's look at, we have another sort of Utube container, and in the center we have a membrane. And it says glucose and sodium chloride are unable to cross the membrane, but water can. Which way will the water flow? So if you notice, both of these are open to the environment, and so they're going to have a pressure potential of 0. So in determining where the water is going to flow, in determining water potential, all we need to worry about is the solutes. Now, because it's part of the same tube, we can assume that the temperature is the same, and we can assume that the pressure potential, or the R value, stays the same on both sides. So in that negative I CRT, all we need to worry about is the I, the ionization constant, and the C, the concentration of both sides. So let's take a look at that. So glucose, the I value is 1 because it doesn't break up. For NaCl, the I value is 2. Which means that our overall concentration on the left is going to be 0.4 plus 2 times 0.5. On the right, we have glucose, which has an ionization of 1, so that's 0.8. And we have a 0.4 molar NaCl. That has an ionization constant of 2. So that means it's going to be 0.8 plus 2 times 0.4. So because the only factors we need to pay attention to are I and C, the overall concentration on side A is 1.4 molar. And for side B, it's 1.6 molar. Remember, that was just the I value times the C value, the ionization constant times the concentration for each of our solutes. So because side B has the overall higher solute concentration, that means it has an overall lower water potential. And that's because the equation negative I CRT determines solute potential. That negative is sort of what flips it around, right? And so the high concentration ends up with a low water potential. So because water flows from a high water potential to a low water potential, it's going to move from left to right, in this case from side A to side B. All right, so that's our last slide for the day. I know this is kind of difficult material, so I really recommend you spend some time thinking about the concepts, thinking about the role of pressure, thinking about the role of solute, how that combines to create water potential, and then try a bunch of practice problems. The more practice problems you try, the better, because that's going to reveal if you have any gaps in your understanding, so that then we can work to move forward and help you really deeply understand this concept. But that's where we're going to leave it off for today.